So as a quick overview of this afternoon's session, it's going to be divided into two parts. So in the first part of the session, I'm going to be reviewing the statistics of our Bayesian decision analysis framework. And specifically, I'm going to walk everyone through the methodology that we actually used for our neurostimulation device case study. But I'm going to try to keep that part relatively brief because in the second part of the session, we're going to be having a panel where we're going to hear from Martin Ho, Kevin Kwok, and Edward Karst, and Professor Lowe about using these methods and patient preferences to design clinical trials. So I'll try to keep everything relatively high level, but I hope this is pretty informal. So if you have any questions or anything's not clear, just please feel free to interrupt by raising your hand. So as we talked about earlier this morning, when running a clinical trial, there are two types of errors that can be made. There's type 1 error, also known as false approvals, and type 2 error, also known as false rejections. And so there can be a difference in terms of the severity of making these two types of mistakes. And so we want to be able to consider this asymmetry when designing our clinical trials and considering the trade-off between type 1 and type 2 error. But in addition to this trade-off, our framework also takes into consideration the length of the clinical trial and the overall error rate. And so what we see is even in the case where we approve an effective therapy, and so we actually make the correct decision, if it takes us a very long time to make that decision, then current patients aren't going to receive much benefit from that treatment. And so we're going to be able to see how we can use our patient preference information in the form of these benefit-risk trade-offs and also in terms of their time preferences to estimate the value lost from a patient perspective in each one of these different scenarios. So let's begin by thinking about what happens when we make a type 1 error, also known as a false approval. And so in our model, in this case, we assume that the investigational therapy provides no additional benefits relative to the standard of care, but it does come with additional risk. And in particular, we identified 3 really important risk factors for Parkinson's patients, at least from their perspective. And so the first is the additional risk of depression. The second is the risk of intracranial hemorrhaging, what we're calling brain bleed risk. And finally, there's the risk of death, mortality risk. And so we can actually use our patient preference information to estimate the relative importance of these different risk factors when it comes to decision making. And so what we saw in our survey results, we found that there was approximately a 6-to-1 ratio between the depression risk to the mortality risk. And so what this means is that a 6% additional increase in the depression risk is approximately equivalent to a 1% increase in the mortality risk. Similarly, we also found that there was about a 3-to-2 ratio between the brain bleed risk to the mortality risk. And so now if we have an estimate of the risk profile of this investigational treatment, we can estimate the average value lost per patient if it gets approved due to patients using this inferior treatment instead of the standard of care. And so similarly, we can also estimate the value lost from the patient perspective due to an error made by a false rejection, a type 2 error. And so in this case, the value lost is going to be related to the missed opportunity to receive a therapy that's more effective than the standard of care. And so in this analysis, we actually classify patients using 5 different criteria that is going to be indicative of their risk tolerance. And so the 5 criteria that we consider are whether or not they've elected to receive DBS treatment previously, whether they experience dyskinesia, the severity of their motor and cognitive symptoms, and finally, whether they have difficulty with mobility. And so now we can see how we can actually use this patient value model to actually estimate the relative severity of a type 1 and type 2 error. But remember, again, our framework also considers the trade-off between the the total length of the clinical trial and the overall error rate. And so we also have to estimate the value lost due to overly lengthy clinical trials. And so to do this, I have to introduce a concept from economics known as the discount rate. And so to illustrate this concept, imagine you're given the following choice. You can either receive one cookie right now, or if you wait for 15 minutes, you get to choose to have 2 cookies. And so the question is, which one of these 2 options do you prefer? And so if you're like me and you tend to be relatively impatient in this context, then it turns out that we can say that you're discounting future benefits in the form of these additional cookies at a rate of 4.6% per minute. And so this is clearly just an illustrative example, but we can use time trade-off questions to actually capture the rate at which Parkinson's patients discount future benefits. And so when we did this in our study, we found that the rate at which Parkinson's patients discount future benefits increased with both their age and also the severity of their symptoms. And so we found that for patients with relatively mild symptoms, the average discount rate was about 15% per year. And so that means these benefits are losing value at a rate of 15% every year. So that looks like this. And as the age of our patient population increased and also the severity of their symptoms increased, we found that the discount rates actually increased, and to the maximum value that we saw in our survey, that was about 30% per year. And so now we can actually incorporate these discount rates, and we can incorporate it with the fact that the length of the clinical trial is going to be related to how large this trial is in terms of the number of patients. And so now we can start to estimate the value lost from the patient perspective of having overly lengthy clinical trials. And so putting it all together, we can use our patient preference information to estimate the value lost in each one of these different scenarios. And the final piece of the puzzle is going to be to estimate the probability of each scenario. And so we can use our statistical framework to estimate the conditional probability of a type 1 error and a type 2 error. So just as a quick reminder, the conditional probability of a type 1 error is this area given in red, and it's also known as the statistical significance of the trial, so the p-values that we've been talking about. On the other hand, the conditional probability of a type 2 error, the probability of a false rejection is given by the area in yellow, beta. And so this is 1 minus the statistical power of the test. And so we can use this framework to estimate the conditional probabilities of each one of these scenarios happening. And moreover, because we're considering a Bayesian framework, we can actually incorporate our prior belief before we even run this clinical trial that we believe that our investigational therapy is in fact ineffective. But if we don't want to incorporate prior probability, we can simply set this prior information, we can set this prior probability to 50%. So putting it all together, now we can use our Bayesian decision analysis framework, and we can optimize over the number of patients in the trial and our decision threshold, which is going to be related to the p-value, to maximize the expected patient value. And so again, I'll review the results. And so what we did is we implemented this framework using patient preference information to design a clinical trial for a hypothetical neurostimulation device. And so in particular, in our trial, we assumed that we used motor function as the primary endpoint. And moreover, we calibrated the benefit risk profile of this investigational therapy using values in the literature for deep brain stimulation. And so, for example, if our therapy is effective, we assume that it would provide an additional on-time of 4.6 hours, but this benefit would come with additional risks in the form of depression risk, brain bleed risk, and also mortality risk. And again, we also ran this analysis for different subgroups of patients. And so, again, we had 5 different attributes within which we classified patients. And so 2 to the 5 there was 2 to the 5 different possible combinations, and so 32 different subgroups of patients that we could consider. And so we ranked these subgroups in order of their risk tolerance, and then we estimated the optimal clinical trial design for each subgroup. And so what we found is that for our more risk-averse patients, the ones that tend to have relatively mild symptoms and have chosen not to use DBS in the past, those patients are on the left. And then, all right, this doesn't work. Our patients with more risk tolerance and relatively severe symptoms and who do have experience with DBS treatment, they're on the right. And they tended to be the most risk-tolerant subgroup of patients. And so we see for this subgroup of patients, the optimal decision was to keep these clinical trials relatively short so that new therapies can be approved quicker. And moreover, the type 1 error rate was set to 9%, greater than the traditional value of 5%. And then on the other hand, for our more risk-averse population of patients, yeah, question?
Are you including people who had DBS before? You are including people who had it and didn't get a brain bleed, didn't die. So what would happen if you took away and did not include people who've had prior DBS? Why did you include them?
So people without DBS are indicated using these yellow dots. So we actually included them as well, but we also want to capture the preferences of this subgroup of patients that does have kind of prior experience with DBS. So we actually considered all possible combinations. But it just turned out, and as Brett was mentioning, that patients that do have prior experience with DBS, and maybe it's because these are the patients who had more risk tolerance, that's one of the reasons why they chose to receive DBS. They tend to have higher risk tolerance on greater than kind of the average Parkinson's patient.
So if I just might amplify that, and just remind me that Dr. Sherman mentioned that we saw pretty much similar features in the obesity patient preference studies as well, that as patients have more familiarity with the devices that may increase the risk, their risk tolerance for those devices were greater. So this is consistent.
You know, I realize the risk tolerance would be greater, but my question would be, are you then, I'm not going to be able to find the right words, but are you sort of loading it up to make it look better?
So biasing the results, yeah.
Are you biasing for including those patients?
Right. So the one thing I would say in terms of bias, if you look at kind of the risk profile of patients who actually do receive an adverse event, while it's substantial, it's definitely by far the minority. I think on the order of a couple percent. So if we included those patients as well, if we still looked at the average DBS patient, patient who had received DBS, I don't think it would actually change the results too substantially.
Show me.
Oh, yeah.
I just want to add that in the model that Sho had mentioned, the DBS experience is actually one of the characteristics that we would be able to adjust for. So in other words, if we are applying the preferences for patients who are DBS naive, we can do that using the model.
So I don't know if this is a question of the order of presentation or if it's actually the order in which you worked, but the way I'm seeing this here is that you have information from the patients on their relative preference for different therapies, and then you presented saying, okay, well, here is the estimate we're using for how effective those therapies are. So when you were working with the patients to determine their preference for therapy, was it for that constellation of fixed effects, or were they perhaps considering in their minds some other range of effectiveness of therapies that might be out there?
Right. So as Brett mentioned, I think we were using kind of 50% improvements in the survey design, but then we do have kind of this model where we run a regression analysis to extend those preferences to different risk-benefit analyses. Does that answer the question?
Yeah, well, I think for that I'd probably have to see more details about how the preference model is built up, because I'm sort of curious, wondering if persons, especially where they are on the spectrum, might feel differently versus for something that is slightly helpful versus something that was very helpful.
Right. And so that's actually kind of in the future iterations that we're going to be looking into this model, looking at things in terms of marginal diminishing returns and things like that. So yeah, that's a good question. And so finally, just to kind of wrap up real quick, what we found is that the fixed significance level of 5% doesn't necessarily maximize patient value. And in particular, for our more risk-tolerant subgroups of patients, the value ended up being lost due to lengthy clinical trials that were too conservative and overprotective of the type 1 error rate. But on the other hand, for patients with less severe symptoms that do tend to be more risk-averse, especially in the case of a neurostimulation device, which does have a substantial risk profile, the traditional thresholds of 5% are too permissive. And so on that note, I'd like to conclude. And now what I'd like to do is open it up to our panel session to get their perspective on using kind of these methods and patient preferences to design clinical trials. So I'm going to introduce everyone from kind of Kevin down to Andrew. And then they're going to provide just a couple, their perspective about kind of this project. So Kevin holds a PharmD from the University of Michigan and has been a biopharma industry executive for 30 years. 9 years ago, Kevin was diagnosed with Parkinson's disease, and 5 years later, he elected to receive DBS, deep brain stimulation, at Stanford. Since implantation, he's been involved in brain radio and closed-loop DBS research. He's also an active patient advocate for Parkinson's and is involved in several Michael J. Fox initiatives, including the Parkinson's Progressions Markers Initiative, the Fox Insights Steering Committee, and the Parkinson's Foundation's Advocates and Research Program. He's also a board member for the Davis Phinney Foundation and is an advisor to the Critical Path Institute. So please join me in welcoming Kevin.
I guess I was the subject of your conversation show that likes to eat the cookies early and not wait. I can tell you that all of the factors that you talked about, I'm smiling at, because the issues of depression, bleeds, were all things that went through my mind, as well when I elected it. The interesting thing is, I think on your graph or the schematic, I'm a bit of an outlier. I was not that severe with my Parkinson's disease, and at the time I had, the biggest nightmare patient of the world, right? I bring clinical research that I Google, and I bring it into my physician, and I showed her this paper on early intervention with DBS. At that point in time, I only had Parkinson's unilaterally in one side, but somehow this sort of geekiness in me said, I'd like to be my own case control. I'd like to see progression of disease, from my left side to my right side. And the outlook of the disease for me was, I knew it was what was in store, and I was not really willing to wait for the iceberg to hit me, 10 years from now. So I elected early on to get involved with DBS, maybe against the wishes of many people, including my family, and even most of my physicians. But being involved in this early stage of DBS, and so, by the way, I've been involved in a 5-year trial that's ongoing right now. So again, the outlier of severity of symptoms and length of study. 5 years is a long time. It's hundreds of hours a year to be involved in a clinical trial. It's called the closed loop trial, offering the promise of DBS on demand. So it's self-adjusting DBS based on various brain patterns. And what it does is it allows it to adjust. I'm currently involved in that trial. I've been the first patient who's actually, been on that. And I feel like for me, it's just my ability to give back. So anyway, enough of my story.
Great. Thanks, Kevin. Next on the panel, we have Martin Ho. Martin is the Associate Director for Quantitative Innovations at the Office of Surveillance and Biometrics in the FDA Center for Devices and Radiological Health, CDRH. He has led CDRH's quantitative innovation program, which turns novel types of information, such as patient preference information, into tools, and then trains reviewers to use these new tools for regulatory decision-making. Mr. Ho represents CDRH as its voting member in the research-involved Human Subject Committee, which is FDA's intercenter IRB. He's also the past president of the FDA Statistical Association and is the chair-elect of the American Statistical Association Medical Device and Diagnostic Section. So please join me in welcoming Martin.
Thank you, Sho. I think your explanation of the result is excellent. I really appreciate how much work you have been doing and trying to improve patients' quality of life through science. And I also wanted to say that our center's commitment to this project is huge. We have the director of neurological device. Here is Dr. Carlos sitting at the back, and we also have the director of clinical trials who just left the room. But I would say that our center director has been very focused on trying to see how we can leverage better use of information to improve and to make to help us to make better regulatory decisions. I would like to say that there are 3 points that I think this project is really impressed me. The first part is that the transparency that we can bring to our decision making through this modeling. As Dr. Shuren had mentioned in his presentation, he said that we used to do this kind of balancing of different conflicting factors in our gut. But now we have a tool that we can help to communicate with different stakeholders to talk about the acceptable level of uncertainty. And as we all know, patients suffer or pay differently for different type of type 1 or type 2 errors. And in different clinical contexts, the relative price between the two differs. So if we can use a tool such like that, such as the Parkinson's disease project, we can help using this tool to communicate our concern from a public health perspective and patient protection perspective. So therefore, I think this tool, the transparency is very important for us to consider the future use of this tool. The second point that I think is very important is from patient preference elicitation perspective, I believe, as far as I know, this is the very first project that we not only are considering the relative importance of benefits and risks, but it also combined with a time discount factor elicitation. And so I think all by itself is actually a breakthrough. And needless to say, we are trying to use these factors in a highly focused way in helping us to attach certain values to different moving parts of a clinical trial design, such as patient recruitment, how large is the patients at large, what is the medical needs for these patients inside the clinical trials and outside, and also what will be the waiting time for us for the enrollment. And I also want to thank our neuro divisions. They are providing all the supports that we need to provide the parameters that you just explained, which kind of capture, in essence, the typical type of device, the parameters that we've seen. So I think we try to do our best to evaluate it as a serious regulatory tool to see how we can learn from it and then polish it and further improve it. So I think I would say that our center's commitment to this project is, to me, really is impressive. And the third part, I think, is that the way that these projects, the conversations that this model can bring up is very, I would say, academically stimulating. Ted, who just asked the questions in the back earlier, he actually came to me last week in another statistical meeting to tell me, to tell me about his opinion about this project and the tool, and how he thought would be able to improve the tool. So, I mean, when before we came here, yesterday we actually we went to Division of Biostatistics to present the same models. And I also received equally enthusiastic and large number of questions that I think they are all very good questions. And I want to emphasize what Annie Saha had mentioned is we are at the early stage of getting this tool, but now we have this tool, we got to learn how to make it operational. Mm-hmm. And, but just like anything else, without the machine to tinkering with, we would never know how to make the machine work. Mm-hmm. So I think it's a very important first step. And I also wanted to say that it's a very wonderful process for both learning and implementing something that can improve upon and communicate different dimension of moving parts when we are designing a clinical trials. Thank you.
Thank you, Martin. And so the third panelist that I want to introduce now is Edward Karst. Edward is the Director of Clinical Research in Neurological Therapies at Abbott, where he's responsible for clinical studies of deep brain stimulation for Parkinson's disease and other conditions. He was previously a scientist in the Real-World Evidence Group and has been involved in early feasibility studies and implanted device software the past 12 years at Abbott and its predecessors. His work has been published in journals such as Neuromodulation, Circulation, and Science Translational Medicine. Edward is also an inventor and co-inventor on 49 US patents. So welcome, Edward.
Thank you for inviting me to this panel. I really appreciate the amount of work you've put into this and the quality of the work. I think that everyone agrees that there's a tyranny of the p-value of 0.05. And it's going to be an uphill battle to convince clinicians, among others, that studies can be designed that have a different target than 0.05. I just recall seeing examples of tables showing comorbidities of patients enrolled in 2 arms of randomized controlled trials and looking at the comorbidities and people looking at one that says 0.051 and saying, well, there's no difference, and looking at another one that says 0.049 and saying, yep, there's a difference there. And that's not the way the p-value is meant to be interpreted. And so there will be some work to do to incorporate this. I also think that from an industry perspective, manufacturers may not be eager to do the upfront work to come up with the data on patient preference. And there's a learning curve for companies, I think, to see the value of it. When a company undergoes a pivotal trial, I should say I'm new to this role. I've been in the real-world evidence group for many years, but I'm just starting to think about clinical trials. But when I think about a pivotal trial, you use data from feasibility studies, from prior literature, and from related devices to make decisions about sample size. But there's often a lot of uncertainty. And you showed a list of assumptions there that were very reasonable assumptions, but it takes a lot of work to get to that point. And there has been many years of experience with deep brain stimulation for Parkinson's disease to be able to have that kind of confidence in those assumptions. If we think about newer therapies such as, a couple areas where deep brain stimulation has been considered are for treatment-resistant depression and for Tourette's syndrome. And there's very little data. And some of these patients presumably have a much different benefit-risk threshold. For example, if a treatment-resistant depression patient is undergoing electroconvulsive therapy on a regular basis, an implanted device therapy might be more appealing to that patient than the population as a whole. So you could think about a lot of these patients may have a different benefit-risk trade-off, but that kind of work needs to be laid out in advance of the study rather than at coming to the end of the study and coming up with a p-value that's greater than 0.05 and saying, maybe we should just do some patient preference work and, not forget that the p-value wasn't what we said we were hoping for. So I think there are a lot of important issues here, a lot of issues that companies will be thinking about in the future.
Great, thanks. And so last but not least on our panel, we have Andrew Lowe. Andrew Lowe is the director of the Laboratory for Financial Engineering, and he's also a professor of finance at the MIT Sloan School of Management. So actually, I'd like to pose a question to Andrew, and this is something that Edward brought up, which is how do we actually approach the cultural change necessary to actually adopt some of these innovations and kind of get away from using a standard p-value of 5%?
Well, I guess I would respond by first asking the question, why is it the case that we've adopted this kind of standard? Why is it the case that we all rely on this 5%? It's not just in the biomedical community. It's true across the board, including in the financial community. And I think it's because it's easy, because we've done it before, we can point to people that have done it in the past, and there's very little risk in using that kind of default. I think the way that we start getting around that is by asking what more can be done? How much better can we achieve results for patients if we start thinking about it in a more systematic way? And I'm going to just use an example that was brought up by Dr. Kwok in his question earlier, which I thought was a fantastic question, about how people feel when they're involved in certain kinds of actions that lead to bad consequences. You mentioned that you felt guilty about encouraging people to participate in phase 3 trials because in those cases some people died. And I want to respond in a few additional ways that responds to Sho's question. First of all, people are going to die anyway. And so it's important for us to think about the best uses of those deaths for posterity. How can we make sure that those deaths are not meaningless, but they actually ultimately end up achieving a purpose? But the other thing I wanted to point out is a behavioral aspect that has to do not with the patient perspective, but rather with the FDA reviewer's perspective, which we haven't really talked about up until now. So, Dr. Kwok, you felt guilty about helping people, encouraging them to participate in trials. Let me ask you a question. How do you think the FDA reviewers felt after those deaths occurred? How do you think it felt for the FDA to realize that they made a decision that ultimately led to the deaths? I can't imagine what kind of moral burden that places on the individuals at the FDA. I have tremendous respect for people that are willing to take on that responsibility for the rest of us. I would argue that that kind of moral burden is an unfair burden to place on anyone, and that burden is exactly the me hurt you burden. You know, on that example, the second moral dilemma that I gave, where I suggested that you push somebody off of that overpass, I suspect that that's exactly how FDA reviewers feel after they discover that a decision that they've made has caused the deaths of people that you can name in the newspapers, and you know their families, and you know their histories. It's a terrible burden. And so in order to deal with that burden, I think that putting together an objective, transparent, systematic, rational, repeatable framework where all of us stakeholders who are in it together decide together what the appropriate values are for approving or not approving a therapy, where we all have input and we are all party to the decision, I think that that's a somewhat more defensible and more sustainable approach to dealing with these really difficult decisions.
Dr. Lowe, I totally concur with your comment on that. You know, I think we mentioned in sort of the breakout session that there's a third option, right? You know, instead of pushing someone out on the train tracks, what if you pushed yourself on the train tracks? And there are a lot of people that might actually say, I would push myself on the train tracks if not only could I save those 5 people, but I could save my children, right, in the process. A lot of these volunteering for clinical trials are you know, my view is that while I'm very enthusiastic about research, you know, once you have Parkinson's as a diagnosis, your neurons are pretty much fried at that point in time. And so the cures are not necessarily for us. Cure is a strange word, right? And that's why I brought up in the Q&A session, slowing of progression is also something that I would think about.
Can I follow up on Dr. Lowe's comments just quickly? I think it's inherently important to talk about this, because you're completely correct, and it's exactly that moral dilemma that drives FDA reviewers to be a little bit risk averse. But if you take the counter side, and I will be the first one here, and I'd love to hear from Carlos and from Owen as well as Martin, who are making these decisions, because I fortunately am not in that role. But when I came to FDA 5.5 years, 5.5 years ago, I recognized the same thing, and I began to talk to lots of reviewers and got different ideas. If you make a positive decision to approve a product and you learn that people are adversely affected, you take that extremely personally. But if you make a decision not to allow a product on the market and people are also hurt, you tend not to take that personally. You don't know about it often. You don't know what would have happened had that device been on the market, because it wasn't. And so, because you don't take that so personally, that also allows you to be a little bit more risk averse. But I think that risk is equal to the risk of making an approval decision and having bad things happen. It's the same when you don't. It actually might even be worse in many cases when you don't allow a product on the market that could have helped many people, and many people are hurt without it. So in terms of operationalizing, as Martin brought up, what we do next, how do we really engage FDA and the review staff, and how do we explain this to the patients? I've struggled with your math and your models for the last 3-4 years since I saw it, and it's complicated. Unless you really are within that realm, it's difficult to handle. It's probably difficult for the rest of the FDA staff, and it's even that more difficult for patients who are focused on their disease and not on the math. So I think it's important to figure out how we explain this, not just to FDA reviewers, but to patients, that FDA has put this thing on the market with this amount of uncertainty. Do we deal with it with labeling? Do we say this is only for patients who are risk tolerant? How do we handle those sorts of things?
I also wanted to add a comment about your part of questions about how to get away from the tyranny of p-value. I think coincidentally last week we have an AlphaMat statistical conference, and Ted and I sitting on the same panel addressing the very same question. And I think there are two things that I think is worthwhile for me to repeat. The first thing is that we don't have to live with p-value. If we can find a better measurement of something that a yardstick, if you will, then we can use that. The second thing that I want to say about that question relating to this model is that this model, I believe, is flexible enough for us to just not focusing on p-value. P-value is only a demonstrative purpose of measurement. We can use a different measurement that are more reasonable, that are more understandable, and plug it in as an input or as a component of the model, and then it can still work very nicely and beautifully. And as for now that we are dividing up the false positive, false negative type of scenario into a discrete quadrants, but I imagine that if we are not thinking about the discrete events, but a continuous event with different probability, I mean, this model can also accommodate that. So therefore, I think it's flexible enough for us when we are planning for clinical trials. We don't necessarily have to stick with 1 point estimate. We can adjust the distribution in a prior probability of different parameters and check it out and see how it works.
I just wanted to add on that what's been done in the history of development of medical devices has often been to target first to do a clinical study first of the most severely ill patients, and then to go after the more moderately ill patients. And a couple examples would be in cardiac resynchronization therapy, where NYHA class 3 and 4 were first the patients studied, and then it was tried in class 2. Similarly, for the transcatheter aortic valve replacement, it was first used for patients who were surgically inoperable, and then has moved into patients who are potentially also surgical candidates because it's a less invasive therapy. But it seems like all of this has a great deal of overhead because you have to design one study, you have to execute the study, and then you have to go to a different study. And so there's this multi-stage process that I feel like some of this could be combined into a single investigation that considers groups higher and lower risk, and potentially could be considered for approval as a whole, based on the original trial design or based on some sort of pre-specified group at higher risk, rather than an all or nothing decision. I wonder if there's ever been any consideration to partial approval decisions.
So let me add to that. I think that there's another mechanism that one could use in addition to focusing on the most severely affected patients. And it relates to, again, Dr. Kwok's own situation, where even though initially your symptoms and prognosis was not as severe, you nevertheless chose to take the riskier path. So there's an analogy in the financial services industry that has to do with taking on very risky financial investments. So we know that typical retail investors are not permitted to take on certain kinds of really risky investments like hedge funds. The SEC, the regulator for the financial industry, has provided a set of rules that allow certain kinds of individuals to take on those risky hedge fund investments. And typically, these are known as sophisticated individuals. Sophisticated is a term of art in the financial regulatory perspective. And it really has to do with individuals that have a certain degree of net worth so they can afford to lose a certain amount of money, and who are deemed to be knowledgeable about the unusual risks of hedge fund investments. So it wouldn't be that difficult, I don't think, to create a similar kind of a requirement for individuals who want to participate in certain really risky clinical trials. A certification process where if you want to participate, you have to demonstrate to the FDA that you have the knowledge to be able to assess the unusual risks associated with experimental therapies. So in addition to what Edward and Dr. Kwok suggested, I think that having a certification process could deal with a number of the issues that Dr. Sheldon raised in terms of figuring out who would be appropriate for these kinds of trials.
You know, the other group that you have to also provide that certification to are the companies doing the research itself.
Yes.
I think to your point, you know, there's a lot of this. I'm from the pharmaceutical industry before I became a Parkinson's patient for 30 years. And what irks me most about my brethren, you know, in the industry is sort of this loving principle of, well, so-and-so got it approved using this trial. And what you're starting to see is sort of more of the same versus innovation, which really stretches you outside your box of comfort. And so, and the argument that I always hear is, well, we have to do that because of shareholder value. We can't put the money at risk for our shareholders. But I think it's a bit of a cop-out, right? And so I think there's got to be some way we can find a way to educate and incorporate this patient value and actually reward the companies that actually do it. Because I do think that right now, as I'm now, you know, the bleeding heart patient advocate, you know, for my company, I go out there and then my CEO says, well, Kevin, you know, we're about to go into registrational trials. We can't afford to add on more costs for doing this. And so, you know, my response is one day, you know, maybe regulators or, you know, just by saying it's the right thing to do is not enough.
Question just in the back, if I could add a comment with regards to the acceptance of risk. Kathy Blake with the AMA. So I think that within the whole ecosystem, we already have a number of ways in which we handle and acknowledge the existence of risk. So one is the most obvious randomized clinical trial, where we say we really do not know what the outcome will be. But I also think that we manage risk through certification of particular physicians, for example, to be the investigators and to take on, shall we say, the early part of the learning curve. Because we know that with some medical devices that you have to get through a learning curve before you start to get a significant, reduction in the amount or the number of adverse events. One place that I might suggest that FDA reviewers go to think about this sort of moral hazard question, phenomenal book written by an ethicist, Charles Bosk, called Forgive and Remember. And the premise, he followed medical teams, surgical teams at the University of Chicago a number of years ago, and he was able to see in these two very different teams an underlying common ethical approach to errors that were made. And what you might call the fully forgivable errors is when you acknowledge the information does not exist anywhere that could have resulted in the right decision. And so that is the research. And thank God some people are willing to take that on, some companies are willing to take on the risk. The next one is where there are multiple choices and options, all of them kind of about the same, all of them with their pluses and minuses. You pick one, it ends up being not good for the patient, but that still is felt to be an ethical choice that was made. Where it gets really interesting is the third group is that you are new to the field. You're in training, you are given, that's the group that may not know something, but you forgive them, but they better remember it, that there was a mistake. And then the fourth group is when you knew something was wrong and you did it anyway. So if you think about, where most of the work we're talking about here lands, it's probably in the first and second of Professor Bosk's buckets, and it's not in the third or the fourth.
Yeah, go ahead. In the back, sorry,
Yeah.
first, and then we'll...
So, great comments from Kathy. I've got some more related ones, and I feel like Martin calling my name often enough, it's sort of like Beetlejuice or being summoned from the back to try and finally say something back to him here. But if you think about the broad context and the framework that's been presented, what it is providing us a context and guidance on how to make a particular binary decision, right? And I think that from a regulator standpoint, that binary decision is should this therapy be approved or not. But other binary decisions are also very important, like should a patient choose to elect to take the therapy when it is approved? And if we can perhaps broaden the framework a bit more to consider that, yes, this has multiple applications for making decisions, and at the same time think about, well, there are different ways in which information can be collapsed into a metric used to guide that decision. So even though appropriately, the common example used to motivate this is in terms of a p-value, and many people think of that in terms of a single test result from within a clinical trial. But this is an action to guide approval, and so there's nothing built into it to say that that actually has to be based upon a single endpoint. And it's not. You have your full harm score and your full benefit score. But that actually brings up the point that, okay, we're not actually necessarily talking about a p-value per se, it's just a set of actions. And the two sets of axes considered are should we approve or not approve, and then the other two are when you should approve and when you shouldn't approve. Now, should or shouldn't approve is in essence unknowable. We don't have access to the true impact of this therapy. We're just trying to guess that. And so that's an area where you can incorporate prior information over time and say, well, here's our current understanding is how often we think this might be appropriate. And from a purely tactical standpoint, it's more in terms of, consider more than just a weight of 0.5 of no effect versus a particular effect, right? You can easily build that in there. But I believe that the regulator should be in a position where they are making a particular decision as to whether or not to approve a product. But even if they have a framework like this where it's clear what the inputs are, it is not necessarily clear that they should be constrained with certain fixed values of those weights. They could have very different concerns for a population consideration than for any individual patient or sets of patient representatives. And so while it can provide a good framework to summarize how the decision was arrived at, there should be room for discretion on the part of the regulator to say, yes, that was an input, but there might be something else that is not fully captured. So I think if we can view this a bit more broadly in terms of a mechanism to guide a binary decision, not necessarily nested in terms of the language of making a call on a particular endpoint, that also brings up, as Martin mentioned before, this concept of it doesn't have to be a p-value, it doesn't have to be a z-score, it could be a Bayes factor. There could be multiple things that you fit into this. And certainly the math gets trickier, but if you make it accessible so that different audiences can use the metric in a different way to reach their binary decisions, then it becomes very powerful.
Yep, in the front. Actually, does anybody want to respond to that?
Thanks. So I think excellent suggestions from two speakers. I think the first thing, in addition to reading books and how we can deal with the ethical burdens on our shoulders, I think another way for us to use this model is that it not only reminds us, this model helps us to move our focus from the harm that the patients suffer from certain device or medical products to the potential number of goods that we can produce for patients who potentially can benefit from a device. So in other words, when we are using this tool, we are explicitly considering the benefit of patients at large, the patient population at large, instead of just focusing on a small group of patients. So if we consider or think about this explicitly for multiple clinical trials or multiple devices, then perhaps that way, that can actually change I would say, the connection within our brains to think, to feel a bit more comfortable about moving from individual cases to a population health perspective, and vice versa. So regarding Ted's comment, I think I agree with him on the approach that is quite very interesting, and I also think that in our approach of thinking about how to use this model, I think everything is, at this point, I think is still wide open. So, but we are open to suggestions and nice product like you. So please keep it coming.
Yes, in the front.
Thanks for a very interesting panel. I wonder if you could go back to one of your slides where you showed how the patient preference incorporated into the equation. I'll just ask my question. I mentioned earlier I was from a children's hospital, and from a medical legal perspective, there's two patient preferences in most cases. So like if you consent a pediatric patient for a study, you consent the parent, and then the child, depending on their age, might have assent if they're over age 6, depending on the state. So go back a couple more.
Sorry, which slide?
Back. Yeah, so the question comes, just go back to math.
Okay. I'll go
back. Getting there.
Math is kind of spread out.
Cookies, yeah, I think this could be it.
Okay.
Or maybe it might be one before this, but the question is, from a mathematical perspective, how do you incorporate and weight the preferences of two people, the parent and the child? I mean, it's an ethical question, right? But it's also a mathematical question. And how do those two things combine to affect the the outcome. You could imagine, for example, in the bar chart that you showed earlier, you've got a progression of bars down depending on assumed risk for the kid, and then you have a progression of bars down for the parent, and geez, you might have two parents too. Anxious to hear your thoughts.
So I think that's a really good point. And especially with some diseases too, you mentioned kind of there's the parent's perspective and also the child's perspective, but in many diseases, there's also the patient's perspective, and then there's family members, for example, who are looking after patients. And can we incorporate those perspectives as well into the analysis? And so I think from a technical perspective, this model is kind of flexible enough to incorporate that, but it is important to note that it is a kind of utilitarian calculation that we're making. You know, the greatest good for the most. But and so you could incorporate the preferences of multiple stakeholders as well into that analysis. And so you can make it this as broad or as narrow as you would like. But I think from a mathematical perspective and a technical perspective, I think we can incorporate that and we can consider what the weights are. But the question is, what are the weights? And I think those aren't questions that we're going to necessarily get from the math, but it's from having kind of, you know, this input from patients, from other stakeholders as well, and thinking about how to do that in kind of a robust and transparent way. I don't know if anybody else on the panel has any thoughts.
Yes, I think it's a perfect question. One, I would like to refer back to Dr. Shuren's keynote this morning. And he mentioned about, he first mentioned about the benefit risk guidance and then use it as a motivation for the preferences study. But what he didn't tell you is that the benefit risk guidance was actually motivated by a preference study being done among the FDA reviewers at CDRH. And at that time, we have conduct a study and we look at the result. And we thought that it may be very important for us to, in addition to the FDA reviewers' perspective or preferences, we should also add the patient's perspective into the mix. So, and this, I believe that's Dr. Shuren's insight. And I think since then, we explicitly including patient perspective in it is because before we can quantitatively measure patient preferences, the reviewers has to do our best to think in the shoes of patients, and at the same time, we also wear our reviewer hats to try to balance the two. And as for what are the optimal weight and what are the criteria to get that optimal solution, everything is in the gut, was in the gut. But I think by now bringing out the patient perspective explicitly, it may differ from our reviewer's perspective, and also it may differ from caregiver's perspective and care provider's perspective. And by bringing it up, bringing up in the sunlight, it already helped us to think in other people's shoes much more systematically. And as for what would be the optimal allocated weights among them, I think it would be something that yet to be resolved in the future. But I think if we don't measure it, we can never optimize it. So by explicitly measure them, that is the very important first step.
So unfortunately, we're almost out of time, but I think we have time for one last question.
Great. Thank you. Okay. Is this on? Okay, good. So the preferences, I'm a little bit concerned about how we make sure that we get the right preferences from the patients. And the reason for that is the framing effects. So you framed it in your survey as a reduction in current, symptoms. we would get different preferences. We know this from behavioral economics, that we would get different preferences if we framed it as preventing future occurrences. I'm also concerned about using the discount rate from people in the present projecting onto their future, because we know, again, from behavioral economics, that people have more fear about future disabilities, and they weight that quality of life prospectively as worse than the people who actually have those disabilities at a given time. So to operationalize this, how do we ensure that we come up with a consistent method of eliciting preferences from these patient groups?
So I think that's a really great point. And I would like to say that, like what you were mentioning, saving lives is very different if you frame it that way than, for example, killing someone, right? It's very different connotation. And so that can really affect survey results. And I think actually this might be a question that Brett might be able to answer a little bit better than me, but I would say this is kind of a first step. We aren't saying this is the final answer. And so kind of this is a first step in terms of trying to estimate patient preferences, getting kind of these utility functions. And then in future iterations, when we learn kind of, as we learn, we can incorporate more of kind of these effects that we know can have substantial effects from behavioral economics, like the ones that you mentioned. So I think that's a really good point. Yeah.
Yes, I would like to add that please come often and come early and often when you want to design a clinical design a patient preference studies. I wanted to say that during the last 18 months, we have received multiple different types of submissions for different type of devices. And we have accumulated quite a bit of experience and try to work with the sponsors to minimize or mitigate the potential cognitive bias and framing effect. I wouldn't say that we can completely eliminate it, but as Dr. Stewart said, there are always uncertainty. We can do our best to do it a bit better, down the road. But we are open to working with all the sponsors to try to polish this and advance the science of patient input.
So
Great, thank you.
On that note, let's conclude, and please join me in thanking our panelists one last time.
All right, we're going to take a short break and then reconvene, I think, at about 2:05. We're going to try and get everybody back in the room for our last couple of discussions for the day. Thanks, everyone.
So, yeah, in fact, excellent presentation, as always. I think there was very common with our events about the wall p-values already. So that's why TAT is one of the tools within the medical device business. We have been trying to move away from it. But as you have said in the book, it takes a measure to fit in it. So p-value